Hello, good morning, good evening. Thank you for listening. This is a medical cannabis show. I respect the law and I hope you do too. And information provided is for educational and scientific purposes, and it is all fact-checked. And the papers that are being discussed will be put in the description below. And if you're listening back for this, the show hasn't actually got a topic yet. It grabs a topic from people in chat usually, and then we go from there. So you might want to skip forward. I reckon you should skip forward at least 5 minutes, just a guess. Just when you see the screen change, that means I'll be showing some slides or papers or going through something that's relevant to the topic. So there you go. And I'll give it till 2 minutes and wait for a few people to arrive. Excuse me. So it's at 1 minute now. Oh, wow. So 60 blimmin' seconds. I can say a few people have said g'day. Hello to Raptor. Hello to Sunshine. Got a comment. I'll wait till 2 minutes. Another 30 seconds to go. Wonder what today's topic's going to be, or even if it's a topic. All right, 10 seconds. Yes, sometimes it's not even a topic. So if it only goes for 10 minutes, that means there's been no topic today. But that's okay. That has happened for a while, actually. It's up to 2 minutes now. So Raptor says, sowing some mascarpone seeds. Happy Friday, friends. 420 Fred says, hello to everybody. Some mascarpone seeds, eh? Very nice. And I'm just going to have a look and see what mascarpone is. Mascarpone, M-A-R. Seeds. Mascarpone. Oh, it's saying that mascarpone is cheese. Or, it's okay, it's related to something else too. I see. Very, very nice. I hope it's a winner for you. Raptor, Genetic Memory Farms. G'day, g'day. Says hello. Hope everybody's well. That's a few nice words. But today still hasn't got a topic yet. So if anybody in chat's thinking of anything that they've heard through the week that's been pretty cool that we could discuss, or maybe even better, a question, that would be all right. Raptor says it's a UK cheese and Dairy Queen. Oh, very nice. You sort of can't go wrong then, I'd say. Sounds pretty good. Whoa. Sunshine's come up with a topic. Says Sunshine. Fire caused by ballasts. HID globes and LED lights. Prevention and growing safe. Okay. That's I can really elaborate on that quite well because I'm a licensed electrician. So through electrical engineering classes, we went right into safety. And I'm actually also a safety. I don't want to go on about me. Let's go on about the topic. Nice. So we've got a topic. Just say, oh, g'day, Mr. Hamilton. Looking for, if you come in a few times, Mr. Hamilton, and say nice positive stuff, I'll monetize you, mate. But for the present, we've got a topic. So fire caused by ballast HID globes and LED lights. Well, it's to do with quality. So let's get up to a slide on that. Where can we go? Here we go. I'll just press and share screen. It's a fire caused by lights. How does it happen? So it happens from heat, because electricity generates heat, and the more heat that comes out, the more electricity comes out, the more heat that's going to be conducted, and especially if the cables aren't wired correctly, if they used everything at the fine line, which Chinese tend to do, and that's why things are starting to blow up, like as you see here in this one. This is the transformers that are plugged in. So there's too much heat's been generated from them, and then they haven't had a thermal overload cut out for them. So that's why it's just kept on working, and then bang, they've just caught fire. And then it's, if you have correct safety switches on your place that you use, it should work fine. What's it called in the States? Say, not ELCBs, not residual current device, that's Australian ones. Implement install. And anyway, if you've had the correct cabling installed, like this shouldn't happen. I'm going to try not to elaborate too much, because I can really go into great detail here. And that's for the ballasts, so for the things that plug in. So you have to really check the load rating, the amount of amps that come out of it, and or even just see if it's hot. Just touch it. And a good time to test is we're coming into summer in the Southern Hemisphere, so now's a good time for this to happen. Northern Hemisphere, it'll be fine because it's coming into winter. But that's the transformers taken care of, and the lights. So the HID lights, these are incandescent. They don't put out much heat. Well, they do, but because it's a short circuit, that's a resistive wire in there which generates current, which illuminates and doesn't break. But in the HID lamps, they just generate a lot of heat, and that's the lines that you see at the side there, the heat sinks. So they're supposed to be there to generate airflow that comes through and pulls the air out, and it's supposed to conduct the heat away from it. So cheaper ones won't have good heat sinks, and more expensive ones will have them. So that's something to always check for quality of your lighting equipment. And to have temperature gauges around, starting to learn what's going on in your atmosphere, because you might find that's good at one time of the day, might be bad at another time of the day, and by having the temperature gauges, you can easily see that, because your body's not a good indicator for, well, a real good indicator for the hot and the cold, if you know what I mean. So having the temperatures gauges around, it gives you a better indication. What was more of the question? Caused by ballast, HID, and LED lights, and the prevention and growing safe. Okay, LED lights. I was with a few people. I'll get onto the LED. I'll just say g'day to a bit, and we'll continue on with that rag question. Thanks, Sunshine. If you've got any questions, please put them in the chat below, or in the chat. Hamilton says, looking for some things that grow in the hot or humid weather. Okay, that's the way. You can always, epigenetics can help you. So that's why heirloom properties grown in your region are really good for those types of events. Ask more of a question, elaborate a bit more if you want, mate. And I can get on to your question, Mr. Hamilton, after this topic, because it's nearly wrapped up. Halfway through it. Genetic Memory Farm says, Sunshine. It says something about someone else. Hello, Midnight. Nice to see you, Midnights. And Mojo Rising. Ground fault indicators. Thank you. The GFIs. That's what it is. Ground fault indicators. So that's what I was meaning to say about your property or your place of where you're growing with lights. it should have GFIs on the circuit. Like in Australia, it's RCDs or ELC. Yeah, RCDs is a newer one. If you use two fellas, Midnight and Mojo, if you come in a few times and say positive words, I'll give you a little monetizing thingo. All right, back to Sunshine's question. And please put your questions in chat because we're on to a bit of a safety heat thing at the moment, which is really cool. I really like the safety. Safety first aid, dead set. You can't go wrong. So back to LEDs now. So what's the go with caused by LEDs? Well, really, it's to do with the manufacturer and more so the region. So without pinpointing which region, which puts out fine line electrical products, actually it's China. You have to be careful of that because they wire everything so it works to say 20 or say 25 degrees Celsius or 70 Fahrenheit, and then they'll do all their calculations on it. So the size of the wire, size of everything, they try and get the cost down, and that's why things are so cheap. But as to do with quality, it's lacking because they've made it so it sort of works, but not so it lasts, as you already know that. But that's how it functions. And to do with the LEDs, it's well in Canada. Here's an example that when it got legalized a few years back, that the government were going and checking on grows to make sure that they had certification stamps on their LEDs, because quite a few ones that come from China don't, and they're the ones that generally cause fires. And what they do, they don't have proper thermal overloads in it, and the circuitry isn't done by an electrical engineering tradesman that certifies the circuit, who goes over in the maths before it. And you do it on paper, make sure that it electrically works on paper, and then that's not too much currents going into the areas that you don't want, so it's going to potentially cause problems. And it's sound for the industry. Germany does that. China lacks in that. Not to say they don't, because they still manufacture some great products, but that's in general what you want with the LEDs. So you've got to be careful with that. They'll just, they got no overloads in them, thermal overload. So it's just dangerous. And what was a bit more of the topic? Prevention and growing safe. So we're talking about fire. So prevention. So, well, that's to do with temperature and not really airflow this time, because, for a fire to happen, you need oxygen, and that gives it its energy source. So by having things open, it's more just knowing your environment and your products. And a lot of people, if they've got, well, they're setting up large grows, they might put all their ballasts, they usually put all their ballasts outside in an area. So it's not inside where the lights actually are for the heat, because otherwise you're just going to get, in the good HID lamps, they've got thermal overload. So once it gets to a certain temperature, it flicks this little thermal analog metal, and it turns off the circuit. So prevention. So that's how you can prevent it. So I'm still looking at that. So you can prevent these fires by just temperature gauges and quality of products and knowing the products, really. And if you're going to overload the circuits too, because a lot of people will plug them in and just keep plugging them in and think you can just keep plugging them in. You can't. They reach a limit. Like it's 69 meters, the root length, 210 feet for a 20 amp circuit. So in that circuit, you can only go to that length. So if you're putting these power adapters on and lengthening the circuits, that's not designed to do that. So you've got to be careful with how you plug in. That's why all the outlets on the walls and things are designed for that way. So that's why you see in labs, there's a lot of outlets to not extend the leads. So it's a prevention tool. So you might need a sparky to put a few in, or split your circuits up. Don't use everything from one circuit, because places have more than one circuit, that's for sure. I'm still sharing. And growing safe in fire. Well, air conditioning helps, but if you can't air conditioning. You just got to really monitor. That's about the best you can do. You can also have some temperature switches, which will turn on and off, which are handy. So they give you indications on how much things are turning on and off. I've got a couple. I don't want to talk about me. That's about it, I think, Sunshine. So safety, that's really rad. I love safety stuff. Fair dinkum. So good on you, mate. Growing safe. Is there anything else that you'd like me to elaborate on? Please elaborate as well. Or please, stop sharing. So that's good. Ground fault. We're up to, hey, there's Lene. G'day, Lene. Hope you're doing well. Mr. Hamilton says, what would you suggest? Sour diesel. Okay, so Mr. Hamilton said, looking for some things that grow in hot and humid weather. So you have to think, what grows in hot and humid weather? Does Northern Lights or ruderalis? Well, no, because that's cold weather, and that's, well, it might be humid as well, but it could be dry, but it's cold. So the hot weather would be more so, well, a lot of things can grow in hot weather. Depends what heat you're talking about. But equatorial strains are the ones that will generally do well because that's the conditions that they're native to. So the ones, and that's where sativa comes in. So you might want to, if that suits your body, because the two differences, I'm caffeine sensitive, so sativa elevates my heart rate, which I don't like. Gives me palpitations, which I don't like. So if it's sativa suits your genetic makeup, it might be something for you to look into, mate, Mr. Hamilton. And humid weather, well, good luck with humid because you're prone to fungal disease, fungal infections. So you've got to work your genetics probably. If a person hasn't already primed the genes, that's something that you could look into, and you could do that through growing it yourself, and then just a few techniques, which if you look through my videos, I've got some on fungal resistant genetics. So there's a few hints in there that you can do to prime your genes to reduce your chances of fungal infections when it's humid. Because most regions are humid overnight when it comes into the morning time when the temperature low, because temperature is relative to the humidity. So if the temperature goes down, the humidity is going to go up. So it's definitely something full on for outdoors. I hope that helped, Mr. Hamilton. If it didn't, can you put in chat some more information, please? G'day. Oh, here we go. Mr. Hamilton says, they tell me Bruce Banner 3 likes to grow on the edge of the light, but they never say how it grows in hot, humid weather. Oh, that's no good. I presume you wanted to grow outdoors, because that's at a guess. And the humid weather, you're in the northern hemisphere, so you'd be coming into the humidity times, which is cold. So it mightn't be indoor. But I've got no experience with Bruce Banner 3, sorry. Grows on the edge of the light. That just means that the metabolism for that phenotype that person has told you about can handle low light situations. So that's where epigenetics comes into it, and where you can manipulate your own for your own conditions, like heirloom genetics. People have grown that for generations in their regions, and then they've just grow seeds again, and it changes a smidgen in the genetics to allow for the conditions on that site where it just keeps growing to. So landrace strains, that's how they're created, or that's how they evolved through those conditions. So you can manipulate your own genetics if you are struggling as well, mate. GMF says, oh, yeah, bit of information for old Ham. GMF's got some good information. What's he saying? He ran some, oh, yeah. Right now, humidity's off the roof. Very good. It's good, people. I appreciate the help that people give in chat to the other people, and they speak from experience too, like that, where you can't go wrong. It's an experience, so it's terrific. There's so many different grow conditions around. Raptor says that's why I use 2 tents instead of 3. Without I can use the AC in summer safely. Yes, by load shedding around, it's good. And safety. What else has Raptor said? Winter runs I use, well, he uses 3 tents, which naturally heat the room. Ha! Brilliant, working to your environment. That's rad. Because yeah, the HID lights, they put out a phenomenal amount compared to LED of heat. LED do put out a fair bit of heat, but the HIDs are renowned for it. And not only through the bulb, a lot of heat is lost through that, but through the ballasts. So when you combine the both of them, that's a good heating source for in winter. And if you get your low ones, those ceramic metal halides that are, you can get the 375 watts ones. They only draw 375 watts. So that's only 1.5 amps, or in Northern Hemisphere, 3 amps. So that's very little, and you could use that as a heater. So you'd put one of them in for winter, even wherever it's close to your lounge room, I suppose, and you could turn it on and leave it on a timer, and then put your veggies even under it. But yeah, it's a rad heat source. Looks like we'll try and, what else can we talk about? I think Mr. Hamilton, you're just looking for hot and humid weather and genetics. Well, let's talk about that a little bit more. Do you want me to elaborate on that, Mr. Hamilton? Let's go into some slides. So we're going to talk about humid weather. Slides of humid weather. Well, what, genetics? I suppose. Or plant growth and development. We could talk about the stomata and its conditions under the humid weather. Or should we talk about how to prevent problems in humid weather? What would you like to go with? How about we'll try genetics? That's very technical though. Let's have a look, see what I've got here while I'm in here. Breeding. Let's have a look here, see what comes up. If any of these slides are relevant. Oh, right back to the start. Well, this is actually really relevant because there you go. Corn used to be on the left. That's how it first came out. And then through breeding techniques and epigenetics, it was changed. And now today, we see 100 years later that it's been worked on and developed. And there you go. So here's on the right-hand side. You can see it's broken down a little bit better. Teosnete. There's now modern-day corn. Actually, and you know with corn too, there's only 4 species, and one of them is the species that you grow and it's popping corn. So you just scrape the kernels off with a knife and peat them, and you put them in the microwave or your oven or your thing out later, and that's the popping corn. So it's nothing, no techniques to it, it's just the genetics. So by you working them over your conditions in your site each year to year and inbreeding, so backbreeding with a parent. So in other words, you might get the pollen. So if you've got the ones you save, you cool, it might herm out and bang, you've got pollen. Keep it, and you're going to work with that. So you're going to store that well. Look in my videos if you want to know how to store pollen well, my breeding videos, or ask in the chat if you want me to elaborate. And then you can go in every grow. So you'll go and sprinkle it on this corn here, for instance, and it'll grow and produce the seed. And then you'll go and plant that seed. then it'll grow up, and then you'll sprinkle the same original pollen, 1 or 2 sepals, so only just a few little tiny bits, just the balls. So you'll pluck off the plant probably at least 50 to 100, and you'll only need 1 to 2 for this application. And then you'll do it again, and then again, and before you know it, you'll marker select. So you'll see, and your selection will be through size, for instance, but yours will be humidity conditions. So which one grows out in more humid and better conditions? And then you're going to do the same, sprinkle that one, and then same sprinkle. Before you know it, you've got one which can put up with ridiculously high conditions in humidity that's going to work to your environment. And it's of the cultivar that you selected yourself, not one that someone else has suggested for you to grow. There you go. See what else we can go into it. No, pure line. You want back cross. This is the method, the back cross breeding method. So crossing with an F1, so the first generation friar, so it stands for in Latin, with one of its parents of its back crossing. So because you'll have saved the pollen, so that's the parent, and then you'll go and sprinkle it, and then it'll produce a seed, and that'll be the first generation. And the second generation, you'll sprinkle this one, and then it'll produce a seed, and that'll be F2, the second generation, and you just keep doing it, and the recurrent parent should be well adapted and should process. That's what you should do to make it stable. Because the problem with the genetics, the homozygosity isn't in effect. Here's a bit better. Say the first year you'll have your non-recurrent parent. So that's the one that you'll be planting, but this is the pollen that you'll have. So you'll be saving the pollen, sprinkling it on, growing the seed. Saving the pollen, sprinkle it on, growing the seed. And before you know it, you're 4 generations in, and this is for a back cross method for a dominant gene transfer. So you're trying to transfer the gene that can be dominant for humidity, for instance, which I don't know that gene. But that's the pathway that you're looking for. So by you doing it and selecting it, and then these ones, you'll grow them in high humidity conditions, and you'll see, wow, look at this one powering. This is doing better. All right, well, I'm going to induce that one. And that's the one that I'm going to be getting the seeds off. You keep going, and yeah, this is how to commercially do it. But by the time you've done it sort of 4th or 5th time, it'll be pretty much where you want it to be if you've done your selections right. So when you're going through the 2nd, 3rd year generations, that's where you have to select the ones that you want for the gene of interest. So you're looking for the humidity gene or the conditions. It's probably not even a humidity gene, but it's a combination of factors that allow that plant to grow under your high humidity conditions. So that's the selection that you're looking for. I think that sign covers it. What's the disadvantage of us doing this back crossing method? New varieties will not be very superior compared to the recurrent parent. Closely linked detrimental genes may be transferred without the target gene. Yeah. So you can get different genes that go and transfer that you don't want, and they could be problematic ones. Well, you'll have to wheel through that. Another disadvantage, highly time-consuming and labor-intensive process. It's not very labor-intensive. I've just explained how to do it. You just got to store the pollen correctly and then pull it out and sprinkle it. Well, there's a technique to doing that. I've explained that in my video. How you get the anther out from the sepal, and then you cut it in half with tweezers, hold it, and then apply it to the flower. And this is what you do want. So because you're using morphological expression, which is through its growth and through your viewing, you can see its eyes, that's the type of marker that you're using for your marker-assisted breeding program, Mr. Hamilton. So the marker, what is it? It's an indicator to identify or highlight anything. So markers are used in plant breeding for successive breeding programs. So there's 3 different types of markers that you can use: morphological, biochemical, and molecular. So morphological is through, you can see it morphing. It's color, is it stretch, is it size, is it shape, is it wilting under the high humidity conditions, or is it thriving under high humidity conditions? Biochemical is proteins and isozymes, or markers that you can see, stains that are under the microscope. And molecular is through testing, through DNA level electron scanning microscopes. Examples of molecular markers. Well, I'm not talking about molecular markers or even biochemical, even though that's quite relevant, stains, but for this instance, not for stains, that we don't need, not looking for starch or different genes that have been turned on. So the product of marker-assisted backcrossing. So they've gone and done a marker-assisted backcrossing, and they've found a drought-tolerant cultivar that is sensitive, is a high yielding as well. So there's an example of morphological, because you can see for drought, is it going to handle it under low water conditions? You don't need to do it the other type, so you can do this for marker-assisted breeding too. The importance of marker-assisted breeding, well, it's a must if you're into a breeding, because the points they raise here is high reproducibility. There's less time consuming, early detection of traits. Yes, that's a very good bonus. You can just get rid of the rest after you see your trait. Small samples needed. Yeah, you can only do it. You only need to do it out of 2 or 4 at the most, because you can see which ones are changing. And cost effective. Well, it's extremely cost effective from what I've explained. You don't need really much. You just need a fridge. Recessive alleles can be identified. So if the dominant gene are the ones that show their up, the recessive ones are the ones that don't. Without going into detail. I've got a lot of slides here. I should stop it actually and see if Mr. Hamilton said anything. Should get rid of that too. He said thanks. Genetic Memory Farm says, Mr. Hamilton, if in my shoes, I'd think about, oh, yeah. Well, that might be, have I explained enough there? Because it does get a bit boring when it comes into the genetics, because not many people are into genetic manipulation. They're into they just want to bloom and grow it. So let me know anyway, because you haven't said any comments. And if I was to continue there, well, I'll show some slides, but I don't think because I don't know what else to talk about. And there's nothing much is, oh, hang on, there is some stuff. Lene said, Lene likes the African. Oh yeah. Mr. Hamilton says, I've done poison, just looking for something different. Yeah, 3 different kind of the grow. 3rd grow is most there. Yeah, 3 different kind on this grow. Okay, so you're not doing a monocrop. You're doing 3 different kinds. That's good. Try and put some more information, mate, and I'll pinpoint your question. You just want to grow it. I can see you're not into the breeding. All right, well, I'll get out of the breeding bit then. I was going to go into breed a bit more and show different examples, but let's go into the resistance. Into fungal problems. Fusarium wilt, like that one. Fusarium wilt is a fungal. Fusarium wilt disease is caused by several variants of Fusarium fungi. So they're pathogenic, and it just makes things wilt like that. Not very good, but that's from fungus. And if you're playing in high humidity, this is a disease, though, that's in the substrate. So it's not really in the flyless sphere. It's not really a botrytis or a, what's that one starts at the end? Powdery mildew or downy mildew. It's not really, those three are in the phytosphere above the soil or above your substrate. So the way to, if you're going to go into your, I know where I can go with this. If you're going to go into your high humidity environment, fungi reproduce at 65% humidity, water activity level. So if your humidity is above that, which it will be because you're looking at high humidity environments, you've got to make sure that you've protect your genes from getting infected with fungal or bacterial infections. I'm looking for the slide that shows the water activity level with the different moistures. Quorum sensing. It's not coming to me. There's a lot of slides here. I'll have to go to the class. What was that in? These are all the courses that I've done at university, and they're all within the last few years, so they're all pretty recent. So the slides that are from them, you could nearly count on them. What are we looking at? Water activity level, which was in irrigation engineering. Oh no, post-harvest operations. Actually, I've made a slide on this. Let's go to my slides. So going through all them. It's not my slides there. One. Oh, fair income. Here we go. Breeding, no, water activity level. Post humidity, here we go. When you're ready. Oh, Jesus. So this shows the, here it is, over on the right-hand side here. So the water activity level on the x-axis, 20%, 40, 60, 80, and 100%. And on the y-axis is the equilibrium relative to humidity percent. So it's relative deterioration rate. The relative deterioration rate, how fast it deteriorates or how fast it works. So an example is bacteria, which is, I reckon, above 90%. Yeast is above, well, 80, but Saccharomyces is as low as 65%. So the growth of mold and yeast and bacteria is higher in higher humidity environments. So that's what this is really getting to. And in the middle one, what's this? The temperature is down here. This is the relative humidity on this y-axis. On this one is the moisture content inside of maize, of this example. So you can see microbes, insects don't grow when the humidity is very low. But when it's above 90, everything thrives. They love it. Here, from 65 to 90, notice bacteria isn't in here because that's supposed to be above 91 in general. And from 35 to 65, insects grow and microbes do not grow. Isn't that amazing? So if you're going to get above high humidity environments, which is above 65%, you've got to make sure that you've got good genetics, or you've got your IPM or implementations to handle problems that might arise. Post-harvest diseases are more severe when relative humidity is 90% or higher. There you go, mate. So that's one slide. I can help you out with that. What was the other things in here? You've also got to keep in high humidity. You'll always want to watch for your leaves to make sure that they're not doing anything funky. They're not twisting or curling or odd-shaped or spotted or anything like that. Wilting, you've got to really watch for that in high humidity. There's a bit of a knack to doing that, mate. It's it might, anyway, you can grow in every environment. Just how good your genetics and your techniques and microbes are to get you through it. Righto, come on. Let's hoping to see some plant stress. Pathogen response. Here's the pathway that will happen. PAMPs or MAMPs, pathogen-assisted molecular patterns, is activated. And they're the genes that happen on the plant. So if you can prime your genes, it'll quickly allow the MAMPs to happen, microbe-assisted molecular patterns. It'll be recognized very fastly. So how I was talking earlier about working with your genetics, that's how it would happen without going and explaining this in detail. A lot of deficiencies here. Oh, you don't want that one. See that one? You think, wow, look at that. That's just a bit of a drop on the leaf, maybe from when I was doing a foliar spray of some type. That's a fungal infection you've also got to be careful of. That's called septoria. Good. Stress, we've already done that. Trichogramma. Trichogramma is an insect, a entomopathogenic wasp that deposits its offspring. Oh, actually, we don't need to go into that. That's not going to help on this instance. Exploring. Yeah, isn't that a spin out? Vanilla is toxic for its own plant, and it grows vanilla. Isn't that a spin out? Just how plants interact, because they can hold. They got these different sections, the cells in the plants, where they hold the different poison, so to speak. It's cool. Oh, that's the last one. Okay. Oh, I should have been sharing for a while. Should see what's written here. Oh gosh. Sorry about that. Get a bit carried away. I like microbes because microbes are the rulers. They fix everything. They can control everything as well. Oh, hang on. Raptor said something. Extremely cost-effective when you consider time and all the expenses, traditional breeding methods. Exactly well said. Yes, for the little bit of hassle that you've got to do by collecting those pollen sacs, the sepals, and going on the sprinkling technique, it's so worth it down the track. I totally agree. Thank you. It gave me some ideas. Oh, excellent. Thanks for the nice words, mate, and feedback. Mr. Hamilton says, I'm trying to breed a gorilla grow, drop, and go. Okay. Very good. Hope it works for you. Bud rot was terrible this year. Ah. Well, I understand a bit now. Yes. You also want to, well, think about what time of year you're harvesting. It was probably too late. So you can look at introducing, this is genetics again, and it's from Lasqueti Island in Vancouver, in BC. Time warp. It was created there to finish early. So if you get hold of those genetics and do the same thing with back crossing with the parent by collecting the pollen, for instance, and then just sprinkling on and then doing some selection on how it's finishing. This one will take a bit longer because it's seasonal, but it finishes earlier outdoors. Cut a long story short. It's really, really cool. So instead of finishing in May, it'll finish in April. No, sorry. Instead of finishing October, it'll finish in September. And that's true. Also, you can consider your position that you're actually putting it in, the outdoors. And if you want to finish a little bit earlier, don't do 180-degree sun. Give it a little bit of shelter, either in the morning or the afternoon, like a decent shelter. So the sun's pretty much blocked off, and that also can help for finishing early, because it sends it in early into flower, gets the florigen hormone produced earlier, and can turn it. Oh, it says, Mr. Hamilton says, I used to do good with skunk, but it's a dead giveaway. Sunshine's outie. See you, Sunshine. Hope you have a good day, mate. Mr. Hamilton says 76% humidity and 110. What? 110 Fahrenheit, for those Australian folk, is 45 Celsius. Quite hot. Did you know that when the temperature is at 100 F or 37 Celsius and the humidity is 100%, we die, or we can't respire anymore. We can't, through our sweat pores, we can't, and it's hard for us to breathe. So plants would be in a similar situation. So, Mr. Hamilton, you most definitely want to work with your genetics, and you'll get some amazing results. I would say if you only do it like you did it this year, if you don't know how to induce one of your plants, you'd do a silver blocking method where you put some sort of silver, a silver SDS, silver disulfate, or colloidal silver, or there's a few others, to spray it on, and then that will block the ethylene, and it'll start producing the male bits, and that's how you'd collect the pollen. So that's how you'd start out. Because if you're in those conditions, mate, that's extreme. It's very extreme. It's streamophile. And you might find that a lot of things mightn't survive in that. You'd far out. That's even around a swamp area, it'd be very boggy because it's so hot. It'd be so overgrown. And judging by the temperature, it's down south in the crocodile alligator area. Yeah, it's full on. What's Lene say? Arizona or Cali, perhaps? Yes. Bloomin' Texas. No, wait, that humidity is way too high for Arizona or Cali. Yeah, it's very high, that humidity. But for getting something to be favourable in those conditions, a lot would take a few generations, because a lot of things aren't suitable for those conditions. That's crazy. Especially in late flower, you wouldn't want to be growing big ones either, because in late flower, when you've got those conditions, on the outside of the plant will be 76% humidity, but on the inside, where there's no airflow, will be up near 90. So everything's going on inside, and that's where you're going to get the problems. So you'd be topping it a lot and doing a heavy prune, even mid-flower, and you'd be wanting to see all the light all the way through it to allow airflow to reduce that humidity possibly. Hey, there's Monty. G'day, Monty. Hope you're well, mate. Yes, hope you're doing all right. Everything's good in your camp. Mr. Hamilton says, this is my third harvest this year. This year? Fair income, that's really good, mate. 20? Okay, 20 inches is 50 centimetres. So he's had half a metre of rain last week. Wow. So the rain factor, if you've got to use microbes that are good for fungicide. they're a fungicide. So they produce pectinase, and that's the enzyme that'll break down the cell walls of the fungi, and that will stop the fungi from growing or spreading. So that's, you want to get into the, also grab some bio-fertilizers as well, mate, and you want to definitely load up with that. If you've got 1/2 meter of rain, there's not much you can do. You can put a canopy up, you can put like a clear tarp up, but it's September area, you're about to crop out in a couple of weeks. You want to prune heavy to make sure that you can see sunlight, and even with the rain far out, you can use vibrators, strigomorphogenic vibrators, and you might attach it to the stem, and then they can shake the plant. And it's an actual induction way, another grow method, but it's a way to get rid of the water droplets. So by just vibrating it left and right, once water sticks on the plant, it'll want to move off, and gravity will help it. So if you're in a heavy rain environment at late flower, it's nearly good luck, mate. It's nearly good luck. Oh, what's Monty say? Everything's going well. That's really good. Got my lactobacillus potassium and phosphorus feed going, as well as a lactose bacillus nitrogen brew. Okay. What type of microbes are you using for that? It sounds like you're only using lacto genus and lactobacillus. Because did you know that a lot of lacto genus, they're not symbiotic, mate? So trying to mix, that's why that's what's used in dairy as a preservative, because it's a heavy biofilm producer that produces a lot of antibacterials in it, and it doesn't allow others to colonize. Just be careful about that. But there's heaps of different ways to grow, and if it works for you, that's excellent. I just like to get into the fine tuning of it. But got the cultures, to have all those brews going, that's rad, mate. That's an organic way of giving mineralized fertilizer. So if you were to have a culture, I've been speaking to it with Raptor about the organic feeds for bacteria like Arthrospira, that spirulina. So to call it organic, like people would like to, they would give it synthetic inputs. But to do it organically, you'd have to do what you're doing. So excellent work, Monty. Mr. Hamilton, and part sun is mostly the trick. Oh, that's a shame. You've got to go part sun to get your outcome, mate. Monty says, can't wait to see the changes with applying these Jadam brews this year. Yes, it'd be good to see the changes for sure. That's Korean natural farming type methods. Mr. Hamilton says, every week, 3 times this week, they've got that massive amount of 20 inches rain. The only thing I can suggest with the heavy rains, if your flowers are too big, mate, you have to shake the plants. Otherwise, good luck. Or you've got to put the bios in there, ones that produce the pectinase, the ones that what's an example of those, he says? Well, trichoderma that's a good fungicide. So you're wanting to source some trichoderma. Pseudomonas florens, that's a really rad fungicide. That's actually that fixes a lot of things. That fixes Pythium. So that's one good, works in the substrates. What else is really good? To target fungus or botrytis or phylosphere, powdery mildew. They even reckon Bacillus subtilis works. That's in everything. That's even in us and beneficial for us. Different strains of that. pretty cool. So yeah, by using your biopesticides, mate, that's going to help you in late flower, and I would highly suggest sourcing those. I'll be selling those down the track. Not at the moment. I don't have those particular ones in stock for you. Monty says, no, it will gas off and kill other microbes, but also have jadams going. Oh, okay. So that's fine. If you're, well, Korean natural farming is a technique that works really well. That's what it's called. That it's got a title to it, and people have grown that way for generations. So it's different ways to grow. So many ways to grow, and to do it, say one type is the best way, is so wrong in my eyes. Even the way that I do it, it works for me, but it mightn't work for other people because they're on different environments, budgets, applications, water levels, all sorts of different things that come into it. So that's rad. That's pretty cool, Monty. It's good to see you getting the different cultures going to do different things for you. I hope it really works out, because I'm doing similar, but with microbes, like just different bacteria and fungi. And I'm mainly focusing on hormones and health. So the microbes that I select have hormones that they produce as well. So it's like giving them a boost of steroids, the plants, all the time, which it's amazing. Absolutely blooming amazing. What happens to an athlete if you give the athlete steroids? They are huge. They pump up in the right conditions, under the right conditions. So it's it's a good area. I'm not going to go on about me. Holy mackerel, 57 minutes. Quick, get your questions in or get your statements in. We've talked about safety at the start, and then we've talked about high humidity growing. I reckon that's actually talked about, only briefly discussed it, because I didn't go into the stomata and different plant processes that happens in high humidity, like the stomas stay open. They like it. There's but anyway, there's no real questions asked for that area, so I just try and focus on the questions, really, and not elaborate too much on what I want to talk about, because that's kind of not why I'm here. I don't want to hear my own voice. I want to help people out from my studies and my experience that I've done. Share that, and we can all share stuff. It's cool. All right. Well, things seem to have slowing up here, and I think I've covered enough. What do you cool folk reckon? I'm not sure what to say. I think those two topics were covered all rightly. Yeah, I'm rambling now. Help me out. Give me some direction. Because if I don't see anything in very soon, that will be a show. Oh, Mr. Hamilton says, yeah, I use lactose on my beans. It really helps. Cool. Yep. And what works on one plant mightn't work with the other plant, because if you've got like the normal is diploids and monos, and if you've got, that's just the chromosome count, but if you've got ones that are, what's the ones that back in the day? Oh, monos. Okay, remember that, I got to solve what. But yeah, depends what species of microbes you use is culturable or colonizable with different plants. It might just not work on the inside. It might work on the outside or even on the roots. So actually that's a whole nother topic, because there's thousands of microbes. Remember that one slide that I can show that shows heaps? Actually, 3 seconds left. Here you go. I'll show This is an example on how different microbes, so I saw it before, microbes, this one, go, this one. Gee, it's annoying, I hate pressing the button all the time. So this is an example of the fungi are in red and the blue are bacteria. So different layers and different sections of the plant. There's all different types of species and genuses. This actually only shows the genus, doesn't go into the actual species. Oh, hang on, yes it does. Ligoniformis. I'll use Ligoniformis. Actually, use quite a, and so by getting the right ones for the right areas, for your right species of plant, will work. There we go, rad. Raptor says, how effective are cover crops with indoor system? They're extremely, cover crops. If you're doing a no-till, it might be good because you want to grow, for instance, clover, which is a nitrogen, has nitrogen fixing in the root nodules, so that might produce it and you can till that in. But if you're growing something that's not beneficial for your soil or substrate, it might be pulling nutrients out, which could be going into the roots of your plant that you want to grow. So it depends on what you're growing. So legumes are very good, and red clover is actually fixes a smidgen more than white clover. If you're into that type of thing, which I am. Or you can also use, there's another thing called sun hemp, which isn't related to canna at all, but it grows very big. It grows 60 days, it'll grow 6 foot, and 90 days it grows 9 foot. So that's another type of cover crop. So you just got to get the right type of cover crop. But yes, they will work because you can get them to work for you by clover, for instance. And get it, if you're going to get clover, get the inoculated one, which has rhizobium with it. Rhizobium is the one that colonizes the root nodules and nitrogen-fixing bacteria in legumes. And some plants, some seeds aren't inoculated with it. So try and get that if you do get it. And then once you get it, you can get seeds that aren't inoculated and you can mix them in. That's what I've done. Mr. Hamilton says, yeah, they lose, but they will tighten up come winter. This is my third harvest this week. I caught them hard and the bottoms come up blooming. I caught them hard and the bottoms come up blooming. Okay. Righto. Yeah, well, you'd be aiming to, if you've got rain coming up and a lot of it, that's when you want to get rid of your big colders, your big flowers, because that's going to cause, well, that moisture inside of it, it can't drop out of the bottom of the plant because it's been replenished from the top. So it'll be nearly permanently in the 95% humidity, which bacteria, any spores that are in there will germinate. So that's why you've got to have the right type of spores in there of your bacteria or fungi to germinate under those conditions. So if it does get in those conditions, they're going to protect your plant for you. If that helps, Mr. Hamilton. Renee's out. Cheers. Have a good weekend. See you, Renee. So yeah, get prepared for your high humidity areas. So with your genetics and bio-fertilizers, and you can shake the plant. You can put up, you can get rid of large flowers before events, rain events. You can cover it, put up some sort of tarp thing, but it's still going to get humidity behind it. So it might actually worsen it because it's reducing the airflow. But I think there's quite a few suggestions said today. I don't want to repeat them anymore. Hope that helped Raptor with the cover crops. The legumes are very effective cover crops because of their nitrogen fixing capabilities. And if you get the right ones, they grow really low. You can get, actually the clover will stretch. It's funny. So it depends on your conditions on how tall it's going to grow, but it won't reach up past quite a few inches, so it'll be all right. But look into the white and more so the red clover as your cover crop and inoculate it. It comes blue is the color I get when it's inoculated. Here we go. Well, is that it? Is that it? Is that it? If you can. I think so. Standard. There you go. You seem like an all right person, Mr. Hamilton. So yeah, please put your questions in or your statements in now. Otherwise, it's going to end. It's been an hour 6 actually. It's fantastic. So today is going to be on safety at the start, related to fires, and then it was on high humidity growing. Yeah. You're welcome, Mr. Hamilton. Tell your cool mates about this channel. So I like to help everybody who's cool. And even the people who, if they're not cool, just don't be rude in the chat. You can still hang out. I don't mind, because I'm here to help. Because I study at university online, and it's just related to plant, soil science, and microbiology. And it's some of the latest stuff. The courses are written within the last few years, so you can trust the information that's been provided and the slides that I show. But being a science, things do get improved and things do change. So keeping that in mind. But my information is within the last 5 years. So yeah, that's it. Like plant nutrients have changed. They went from 16 in the 60s to 17, then up to 19, and then back down to 17, which they're at today, because they found that 2 of the essential nutrients of the plant are actually beneficial. So they changed the category. So that just shows you about the different science. Thanks for your informative discussion. Good on you, Raptor. Thanks for you being part of it too, mate. I need the tech folk in chat, so things that they can help me in little hard bits and areas that I lapse in, and they can also share their experience, which is vital. So yeah, it's quite a few cool people. Well, not cool, everyone's cool, I think, in the chat, but there's quite a few research type folk and who do experiments so they can share their experiences, because money can't buy that. People can go and study at uni as much as they want, but they can't. Putting that information into practice is a different thing. So experience is vital. So between the experience and the education, it makes for hopefully a good show. So yeah, tell your friends, Mr. Hamilton, and thanks for the nice words wrapped up. Well, with that, I reckon I appreciate everybody for listening and for putting all the words in chat and making it a show again. Thank you heaps again. I'll see you all next week. Happy breeding, happy growing, and good health to you all. Bye-bye. Right. Bye-bye. See ya.